Hello everyone, welcome back to my channel Learn Biology in Easy Way. In this video, I am going to discuss an important topic of zoology, that is role of microfilaments in amoeboid movement. Microfilaments play an important role in the formation of pseudopodia in amoeboid movement. Pseudopodia extend and contract through the reversible assembly of actin subunits into microfilaments and of microfilaments into networks that convert cytoplasm from a sol to a gel. At present, it is known that motive force in amoeba is generated by sliding interactions between actin and myosin filaments in a way comparable to that occurring in muscle cells. Actin in amoeba. Actin is the major structural protein in the force producing event. This contractile protein has been identified in amoeba proteus and acanthamoeba. These actins exist as G-actin or globular monomers and F-actin or polymeric filaments of G-actin units. Like muscle actin, actin in amoeba can reversibly bind with heavy meromycin to form arrowhead-shaped complexes visible under electron microscope. Myosin in amoeba. It acts as mechanochemical transducer in the force-producing event. It has been identified in amoeba proteus and acanthamoeba. The protein has a globular head and rod-like body comparable to heavy meromycin and light meromycin of muscle myosins. It can reversibly bind to actin filaments. This is a diagram of F-actin of acanthamoeba. Electron microscopic study of the thin filaments following negative staining with uranyl acetate revealed that each filament has an undefinite length but a width of 6 to 7 nanometer. Each filament has a double helical structure just like muscle F-actin. Thick microfilaments resembling the myosin filament of muscles have also been identified by electron microscopy from the cytoplasm of ATP-stimulated amoeba proteus. Each filament has a length of 500 to 700 nanometer and a width of 16 to 22 nanometer. Each filament shows lateral projections like muscle myosin filaments. Sliding interactions between actin and myosin filaments. Holberton and Preston 1970 reported that in the glycerinated models of amoeba proteus, the cytoplasm shows thin filaments, but when such amoeba were activated by treatment with ATP, numerous parallel bundles of thick filaments appeared in the cytoplasm. Clusters of thin filaments were aligned in between tracts of thick filaments. Either end of each filament was associated with 6 thin filaments. In this figure, we can see alignment of thick and thin filaments in ATP-activated glycerinated amoeba. Such findings strongly indicate that ATP plays a role in formation of stabilization of thick filaments and sliding interactions between thin and thick filaments occur in amoeboid cytoplasm during the movement. Next, relation between sliding of microfilaments and amoeboid movement. Commoning, Stockham and Botterman 1973 hold that sliding interaction between microfilaments takes place in the ectoplasmic cortex, especially at the uroid or tail. This reduces the volume of ectoplasmic cortex at the uroid. For this reason, a hydraulic pressure is generated in the tail and this causes forward streaming of endoplasm or movement of the amoeboid cell. Factors required for the interaction of actin and myosin proteins are ATP, calcium and magnesium. ATP stimulates the formation of thick filaments and their interactions with thin filaments. Sobota 1985 revealed that the calcium stored in CADD calcium dependent electron dense deposits may be utilized for the activation of contractility of the cytoplasm in acanthamoeba. Here we can see a figure of calcium dependent electron dense deposit CADD on Acanthamoeba plasma membrane. Thank you.